The cell spends 90% of its time in interphase. Interphase is split up into three parts: G1, synthesis, and G2. During G1, the cell grows and builds proteins it needs for cell division. It also takes in nutrients. In synthesis, the cell replicates its DNA so each chromosome has two chromatins. Finally, in G2, the cell grows and builds proteins in preparation for mitosis and cytokinesis. Centrioles are duplicated in G2. In prophase, chromatins are condensed into sister chromatids. Centrioles begin to move towards the opposite end of the cell and start forming spindle fibers. Finally, the nuclear envelope breaks down. During metaphase, the nuclear envelope disappears and the chromosomes line up in a straight line at the center of the cell. The spindle fibers, cast from the centrioles, attach to the chromosomes. During anaphase, the sister chromatids split at the centromere and move towards their respective poles of the cell. In telophase, the spindle fibers dissolve and the chromosomes slowly loosen into chromatin form. The two nuclear envelopes begin to reform as the cell readies itself for division by forming the cleavage furrow. During cytokinesis, which is the final stage of cell division, the cell splits itself into two daughter cells by expanding the cleavage furrow with the use of microtubules.